Hello and welcome to Unit 4. The subject matter for this particular unit will be three specific types of airborne contaminants: gases, vapors, and particulates. Now remember what we've told you so far. The industrial hygienist, in a very traditional sense, is most classically known for doing air sampling. We've also told you that the vast majority of published exposure limits that industrial hygienists use by which to evaluate the data that they gather are established for gases, vapors, and particulates. The vast majority of them are. So it's appropriate that we would look at each of these three categories specifically. So the other thing that I was going to mention that is important at this particular juncture is that when we talk about air sampling, there are established methods by which the industrial hygienist will collect that sample. So the method might say collect 400 liters of air over the course of an 8-hour period of time, use a 2 liter per minute sampling rate, and then use a very specific type of filter media. So for example, for an organic material, we would use a tube that's filled with activated charcoal, whereby the organic molecules actually adhere to the surface of the activated charcoal. We then suck air through for the appointed amount of time. We would then send that tube to the lab, ask them to analyze it. There are, of course, many different types of analytical methods that are used to say how much of a material is on something like activated charcoal or on a filter, for example. And then ultimately, the lab calculates the concentration. It looks at the amount of air that was sampled, and it can give us a overall concentration, which hopefully is as an 8-hour time-weighted average. That would be a very typical type sampling method. So what we learn in industrial hygiene very quickly is that the analytical chemist at the lab that you send your samples to is your best friend when doing an industrial hygiene survey. I say that because they are intimately familiar with exposure limits, they are intimately familiar with sampling methodologies, and so forth. So with the wide range of things that we can sample for, we really rely on an expert like the analytical chemist at the lab to guide us and make sure that we're collecting the material on the correct media and we're doing it in the correct fashion. So again, we take that result that we get and we compare it to an established exposure limit. And then we ultimately have to write up our results in the form of a sampling report. So again, we'll talk about more specifically the idea of sampling later on in the class, but you need to be thinking about the big picture as you look at some of these individual categories of airborne contaminants. And as is stated in the written materials and the text, it all kind of fits together if we think about how a particle is generated or an airborne contaminant is generated, transported to the air, through the air, and ultimately interacts with the worker, and we have an exposure. And the way we sample materials is ultimately dependent on, for example, in the case of particulates, the physics of how the particle behaves in air and even how it gets into the lungs. So, for example, a very small particle at some point would actually behave like a gas inside of our lungs and would be inhaled and exhaled just like gaseous molecules are. Whereas a very heavy, large particle would tend to be deposited and probably very early on in the respiratory system, for example, in our nose, perhaps even in our mouth. So we're trying to give you the big picture, and we're also building each of the pieces of the foundation so that by the end of the course you'll really understand the world of industrial hygiene. So with that, I say once again, welcome to Unit 4, and I hope you enjoy learning more about gases, vapors, and particulates.